There we go. Okay. Welcome, everybody. This is the third installment of the EEID Forum Seminar Series. Welcome to any of those that are coming for the first time. We're just starting here this fall, but we're going to continue on in the spring. And the whole idea here is we're trying to just create a sort of online community to connect people working on host parasite interactions. And we're sort of starting around this online seminar series that we're doing today. And for the current theme, we're sort of focused on climate disease interactions, but that'll probably change over time to other things. Our hope is that maybe this will have other spin-off things that happen at a later point in time. Maybe there could be some student discussion groups that sort of bud off of this that we sort of help facilitate through this sort of online forum. But then maybe other links to teaching materials and so forth. So we're particularly trying to connect people in places that may not have a large community of colleagues that do this kind of host parasite interaction type work. And so yeah, this is just our third time meeting with everyone. If you missed these, then we do have them up on our website. You can look at the past ones so far, and we'll keep posting them there, and we post them up on YouTube also. This is our first time back from US government shutdown. So as a government employee, and for Kevin too, welcome back. Thanks for your service. And thanks to the wider EEID community. I just feel like we got to do that daily affirmation sometimes that we're good enough, we're smart enough. Gosh darn it, we do things that are important. And thanks for all of that. Sometimes it's hard to see whether or not you're having that impact that you want to have. And in those times, I sort of think about the long arc of science and how the things that you do now might have ramifications much later on that you don't know about. So that's everything from Mendel and his pea pods to the early stuff on mRNA to safe and effective vaccines. So thanks for all that work. And so with that daily affirmation, I'll pivot over to Kevin Lafferty, who's our speaker today. Thanks, Kevin, for joining us. Kevin is a senior scientist with US Geological Survey and one of the pillars of the EEID community. So great to have you here. His work's been really inspiring to me and many others. To paraphrase it in a couple sentences, it's sort of showing how parasites and pathogens, although unseen, are hugely important in the structure and functioning of ecosystems. And one of the things that I appreciate about Kevin's work is he sort of has the courage to stir the pot on controversial issues and challenges us to really think about, anything from the importance of how does biodiversity interact with infectious disease to probably today's talk about climate and disease interactions. So thanks, Kevin, for joining us and giving today's talk. And let's just kick it over to Kevin. Thanks so much.
Well, thank you, Paul. And boy, what a joy to see familiar faces and even more so new faces. Wish we could do this in person, but we'll do our best in Zoom. I realized when I told Paul what I was going to talk about it that I probably could have chosen a broader topic and included why and what, but we're going to go through just sort of snippets of studies. And I just wanted to say if any of this stuff is interesting to you and you'd like more detailed or discuss, or you have issues or disagreements with me on particular studies that I'm going to mention very briefly, just reach out to me. I'm always happy to talk about them. I am here on campus at UC Santa Barbara, and in putting this talk together, I thought, I should let people know that. Let's see. I have been on campus at UC Santa Barbara for a long time. And in fact, I came to UCSB as an aspiring marine biologist because on campus we have this access to incredible kelp forests and ecosystems. And I'd wanted to be a marine biologist since I was 13. And one of the things that I'm particularly interested in are food webs in the ocean and the connections among species. So, for instance, here we have giant kelp, which is a foundational algal species in the background of that photo. And then, so that's on that bottom row of that food web, that's the brown dot. And then you see that first line of nodes in that food web, those are all the herbivores, including this school of zebra perch that are literally 100 yards from where I'm giving this talk right now. As you move up that food web, you get to higher and higher trophic levels until you get to really cool things like great white sharks and stuff. So that's always been my attraction and desire of being a marine biologist. Now, as I went through, got a bachelor's degree and then a master's degree, and then I went to work for my PhD in the lab of a guy named Armin Kuris, who's really sort of a figurehead in parasitology. And I made it really clear to Armin that I would work in his lab as long as he understood I had no interest in parasites. I found them disgusting and uninteresting, and I was a marine ecologist, for God's sakes. So that was fine, except for the fact that I wasn't really a great student. I'd had no fellowships or access to funds, so I had to teach every quarter. And one winter, the only course that was available for teaching was the parasitology course. And that sucked. It really sucked because I'd never taken parasitology. I had no interest in it. And there was no other TAs, and there were three lab sections. And I was literally, and I'm sure everybody online has been here, trying to stay 30 minutes ahead of my students and prepping for labs and stuff. But after a few labs, the way Armin sets up his course is we go out and we collect fresh material from the ocean, and lab number two, every student gets a shark to dissect. And it soon became apparent to me that all these species that I thought I knew everything about as a marine biologist were filled with parasites. And interestingly enough, I'd never even seen the word parasite in a marine biology textbook as I was doing my studies. And so I became more and more convinced that not only was there's this very impressive diversity of parasites in the ocean, but also that sometimes they were doing important things. And so, for instance, if you look closely at this photograph of the great white shark, you can see trailing from the dorsal fin the egg masses of these parasitic copepods. And so if you were really to ask who's the top of the food chain out in the kelp forest, it's not the great white shark, it's these guys that are feeding on the great white shark. And in fact, one of my recent PhD students put together the parasites in the kelp forest food web, and you can see that they are the top of the food web. And not only that, but they're incredibly connected in this food web and comprise a large part of the biodiversity in the system. So that is kind of what got me interested. And so I'm going to, before we get into the climate stuff, I'm going to talk about just sort of global changes in general and how they affect parasites, because I think that's really important to help understand the climate context of things. So if we look at this food web, let's just pick a particular species. It's a video I took not far from here a while back. It's two spiny lobsters fighting it out, and then one sees I'm there and it starts to come over and chase me off out of the picture. But these are a generalist predator. They eat a lot of hard-shelled invertebrates in the system, and they may have important effects. And people, including myself, have studied a very simplified version of the food where lobsters eat sea urchins and sea urchins eat kelp. So where you have a lot of lobsters, you have fewer urchins and you have more kelp, blah, blah, blah. What's interesting from a disease perspective is that we also have diseases that infect these urchins. And so to a certain extent, lobsters and diseases are competing for urchins, and both can depress urchins to allow kelp forest to thrive. One of the basic things we know from disease ecology is that many diseases have these mass action transmission where you have a greater probability of an epidemic when the hosts are really abundant if disease is transmitted through contact, as it might be for this particular disease. So these epidemics are more common where urchins are abundant, and this then instantly brings us into the importance of food webs because lobsters are important in determining the abundance of urchins, so they should have indirect effects on diseases. And we can quantify this by comparing sort of a natural experiment of areas where fishing is not allowed, marine reserves, that's the left-hand column of these abundances here compared to areas that are fished. So outside of marine reserves, we fish all the legal lobsters basically because they're so valuable. And that means we get increases in sea urchins and decreases in kelp, but it also means we get big increases in disease outbreaks in urchins. And so one of the consequences of this, what you might call global change of fishing down food webs, is an increase in sea urchin diseases. Now, you may not care about urchin diseases, and I wouldn't blame you. And it does bring up the secondary question, which is what parasites are really problems? In other words, disease ecology is kind of a problem-solving discipline. So if we were to look at this food web and ask of all the parasites in that system, which are problems, if you start with the perspective that that blue dot in the kelp forest are humans, because we are in the kelp forest food web, then parasites that can infect us are potentially problems, and parasites that infect the things we really like are potentially problems. But it only amounts to 56 out of 444 parasite species that you could claim would be quote unquote problems in any extent. And I just wanted to add that as context because obviously a huge fraction of parasite diversity are not problematic. And yet we can learn some things from them, as I'll discuss later. But lots of infectious diseases are problems and they have impacts, and those impacts are not distributed evenly, or you might say equitably across the globe. Where I am currently in North America, human disease impacts are relatively minor relative to the Southern Hemisphere, and particularly Africa and India, where the disease impacts are outsized on a sort of per capita basis. And we can look at some of the most important human infectious diseases, and we can ask, well, what are the things that are likely to drive those in space and time? And I'm going to come back to this full list later, but I thought I would start with a food web example for schistosomiasis. Now, I'll go over, I think the life cycle is worth mentioning for those of you that might not be familiar, but schistosomiasis is called by a trematode worm that uses a snail as a first intermediate host. Those snails produce these cercarial stages that go and infect humans. The worm then migrates through the circulatory system into the venal system, where the male and female, they're separate sexes. They stay in permanent copulation, sort of like a hot dog in a hot dog bun. And then every day the female lays a bunch of eggs, does a few dozen, 100 eggs, whatever. And those eggs have to get out of the human body, which is not easy if they're produced in the circulatory system. So they cause a lot of damage trying to get out, and most of them don't make it, and they end up stuck in either the liver primarily or for another species of schistosome, in the bladder. They cause those organs to fail or be less efficient and lead to all sorts of health complications. So it's a nasty disease, but it chronically builds over time as you accumulate this legacy of eggs day in and day out. Okay, so that's the disease. We know the factors that promote this disease: poor sanitation, obviously, because you gotta excrete the eggs into the aquatic habitat to complete the life cycle, and also good habitat for snails. And so we have a pretty good understanding of what caused the largest modern outbreak of schistosomiasis in the world in the Senegal River Basin. Basically, the Senegal River, there's not a lot of altitudinal gradient in Western Senegal, and so when the tide comes up the Senegal River, it can go for 100 kilometers, and that means you can't really grow crops or do irrigation along that part of the Senegal River. So the World Bank decided to fund a dam that would block the tide from going up the river. And that did do exactly what they had hoped. It meant that you could irrigate for 100 kilometers sugarcane plantations on the Senegal River, and some of the landowners there made a lot of money. They brought in workers from the south of Senegal to work those fields, and those individuals, many of them had schistosomes in their bodies, so they brought the parasite with them. And then by changing the salinity of the river, they created really good habitat for freshwater snails. And then we had massive outbreaks of schistosomiasis with, most people living along the river becoming infected at very young ages and at very high intensities with consequential catastrophic health issues. Now, we went there as interested in how that dam might have affected things. Now, we know that obviously fresh water is good for snails, but was there more to the story than this? And when we got into Senegal, we started asking around about the food web. And one of the things that piqued our interest was that older fishermen could remember fishing these giant river prawns in the Senegal River, but the younger fishermen didn't remember them. They'd heard of them. And so the thought was that the dam might have interfered with the breeding migration of these prawns. The adult prawns live up in the freshwater reaches of the river, then they migrate to the estuary to breed, and then the larvae walk back upstream. Putting a dam there prevented that migration and caused local extirpation of these river prawns, we suspect. So the reason that this is important is that we are quite familiar that the favorite food of these river prawns are freshwater snails that transmit schistosomiasis. And so we could demonstrate in aquaria, if you put a prawn in a tank with snails, they ate all the snails. The only ones that were left were the ones that were able to crawl up out of the tank and get away from the prawns. And so that led to our proposition that we might be able to reduce schistosomiasis by adding prawns back into the river, and specifically in places where people become infected. So we proposed to do a field experiment focusing on putting prawns into places like this water contact site near a village in Senegal. Now, Senegal is a really hot place, and so if you're a kid, you go swimming every day after school. If you're there, that's where people wash their dishes, they wash their animals, they get their water, and transmission occurs here because it's real. You see all that aquatic vegetation is really good habitat for snails. So we identified two villages and we asked if they would be willing to participate in a study. And the first thing that we did is we got the school kids, had a mobile clinic. We tested them all for schistosomiasis so we could count the fraction that were infected and how heavily infected they were. And then we treated all the kids with praziquantel to eliminate the worms from them. Then in one of the village areas, we built a little barrier that you can see there in the background, and we introduced prawns. And then we tracked how things changed over time. And by tracking the snails, what we found is that where we introduced prawns, just like in the aquaria, the snail populations were reduced, especially the populations of infected snails, and that was important. But more important than this was bringing the school kids back 6 months later and then checking them again for eggs to see who had become reinfected. And what this figure shows is the two villages. The purple village is the village that initially had the most intense infections. We treated both of those villages, and then for ethical reasons, we put the prawns in the village that had the most intense infections, and then over time, reinfection rates were much lower for that otherwise heavily infected village. So we were able to find that by restoring prawns to this river food web, we were able to potentially improve human health outcomes for these school kids. Now, I wish I could say that we've solved the problem in Senegal, but as with many things, the little hiccup here is that the prawns really like good water quality, and the water quality around these village sites is really bad. Like it's polluted with soap and pesticides and stuff. So the prawns don't stick around if we don't put the barrier net in. So we haven't found a super sustainable solution there. But as far as proof of concept of the importance of restoring food webs to bring human health, I think it's an interesting example. I want to stick with this type of parasite for a moment. It's actually the system I've worked with the most in my career. Again, trematodes that use a snail as an intermediate host. In this case, a whole community of trematodes, 18 different species that have this life cycle in our local estuaries. Each of those species has a slightly different life cycle, and many of them use not just a snail, but they use a second intermediate host as well, a fish or a crab or a clam, depending on the species. And so the thing that we were interested in was if the food web is really complex and it has a diversity of species in it, especially these second intermediate hosts, does that create more opportunities for different trematode species to complete their life cycles? And so the aspect of diversity of the system that we were interested in looking at was the diversity of parasites that we see in snails and the diversity of birds at a particular habitat within the marsh. So we went to the different sites within the marsh, and you can go to the mudflats there and pick up 100 snails in about 5 minutes, put them in your pocket and take them home and dissect them and figure out the diversity of parasites at that particular site. But to get at the birds, if you walk out with binoculars, you scare all the birds away. And so we went with time-lapse videography that takes about clips of video every minute for a couple of weeks. That gives us 2,000 clips that we can then show to undergraduates who are very pre-adapted to looking at video clips. And then they can flag the video clips that have birds in them, and then we can go through afterwards and identify the birds and figure out, what the birds are doing. Are they just passing through, or are they feeding? The videos really work nicely at high tide, where it's not that convenient to go out and survey ourselves. And then the video cameras don't disturb the birds, so we get a really high accurate picture of what the bird diversity is at a site. And then, as you might expect, if you plot the diversity of birds at a particular site against the diversity of parasites at that site, we get a really strong positive correlation. So more birds, more different types of life cycles could be completed, more different species of parasites are able to persist at a particular location within that snail population. We could sort of validate that with a quote unquote natural experiment, because next door to the nature reserve where we work was a degraded part of the estuary. And you can see this figure shows the diversity of parasites in the nature reserve in the dark bar compared to that degraded area in the light bar. And we initiated this study because the city of Carpinteria was going to spend $1 million to restore that degraded area. And we wanted to see what happened to parasite diversity after restoration. And I will say that the first year after the restoration, parasite diversity crashed, because they brought in all this heavy equipment and they made all sorts of disturbances. But after 6 years, the parasite diversity really increased in that degraded area, and in fact exceeded the diversity of parasitism in the nature in the reserve where things were already high quality and diverse. So we had a meeting with the city, and we congratulated them on spending $1 million to restore parasite diversity, which they did not find humorous until we explained to them that the only way those parasites were coming back to the system is because they had done a good job of creating habitat suitable for a diversity of fishes and invertebrates that then provided an attraction and food base for the birds that came into the system. The relationship between host diversity and parasite diversity, we sometimes call it host diversity begets parasite diversity. And it actually has some important mathematical details, because the response of parasites to host diversity really depend a lot on how complex the parasite's life cycle is, and how many host species the parasite can use. And so basically the rule of thumb is that species that are very specialized and have many hosts in their life cycle, they're much more sensitive to biodiversity loss than species that have a single host and are generalist in the types of host species they can use. And so that leads to a variety of relationships between free living richness and parasite probability of being able to occur there. And that's what all those different lines show, of different host generality and different life cycle complexity. On average, when you take a food web like the ones in our estuaries and you average them all together, you find that it's almost this weird one-to-one line of, if you reduce free living richness by 50%, you reduce parasite diversity by 50%. That's pretty much how it averages out. And that leads to this general question, which is a healthy ecosystem one that is rich in parasites? It was sort of a controversial topic at the time. And I wanted to give you one more study that suggests, how that's really sort of obvious. And that has to do with working in Arctic lakes up in Norway with our Norwegian colleagues in Takvatn. We're able to put together another food web with parasites in it. There, the parasites are in red again. You can see they're lined up on the right-hand column there, and the free-living species are aligned as rows with black links among them. And this is what the food web looked like before 1920, when the Norwegians got tired of not catching fish, and so they introduced Arctic char and stickleback to this lake. The lake had already had brown trout, but they'd fished all those out. And what happened was, by adding these two species, by increasing the fish diversity in the lake, they created opportunities for 5 new parasites to complete their life cycles in the lake. And so the whole system became more complex due to these new opportunities of new hosts that the parasites could explore. So there's a lot of evidence for the fact that biodiversity creates opportunities for parasites, which is slightly different from the more familiar concept of the dilution effect, which talks about how if increasing biodiversity increases the relative abundance of non-competent hosts, then you can get the reduction in the prevalence of a particular parasite species. So that's related in some sense to this diversity effect, but it's not about diversity, it's more about prevalence. And so this is just an example of how you can get a dilution effect in the Lyme disease system. But there's other ways that you can think about connections in a system, for instance, through the effect of increasing forestation and how that might affect a key host like deer, that then might increase the abundance of tick vectors and so on. And the idea here is that if we consider the additional complexity and put in maybe a more complex path diagram to ask how changes in forestation might affect infectious disease risk, we can see that it really is less simple. It's not necessarily obvious which way it's going to turn out. It depends on the relative strengths of these different paths. You can get dilution, you might not. It's a context-dependent issue. Now, a lot of the the studies on diversity and the dilution effect have been done in a fairly small subset of systems like Lyme disease. And we were curious about how these predictions might apply to a much wider range of the globally important human parasitic diseases. You should read every line in that table. But the point is that we can ask, based on the transmission mode and the parasite's interaction with different host diversity, et cetera, what are sort of our a priori expectations for how an infectious disease might respond to changes in biodiversity. When we tabulate all that, we find that basically a lot of different outcomes are possible, but a very common outcome is no effect. In other words, a lot of infectious diseases of humans really don't interact with biodiversity all that much, and so we might not expect them to respond to changes. And there are cases where biodiversity loss should lead to more disease and cases where biodiversity loss should lead to less disease. Of course, those are our a priori hypotheses. The real key is to go into these systems that have been not studied and do the empirical work to find out whether there's support for these hypotheses or not. Which finally gets me to our favorite, this is like our icon, this should be on all of our t-shirts, the disease triangle that defines every introductory lecture to disease ecology. And I started getting into this back in basically before disease ecology was a thing, back in the mid-90s. I did this, if I'm generous to myself, I like to call it a meta-analysis, but we weren't doing meta-analyses back then quite. This is a vote tallying table of different studies that looked at different types of stress and the response of different types of parasites. And basically, if the papers all said that the parasite increased in response, they got a plus. If they all said it decreased in response, they got a minus. And you can see that basically the upshot is there's lots of pluses and minuses, so there's no universal response of parasites to stress, but there are some themes. So some parasites tend to respond positively to a wide range of stressors, and some stressors tend to increase a wide range of parasites. And then also there are negative effects of stressors on parasites that have some patterns. But the one that really struck me was the thermal effluent. That's that second column that shows generally positive effects on parasites. And these data are mostly from aquatic systems at the time. And the reason that was of interest to me was because Armin Kuris was on sabbatical, and so I was teaching the parasitology course. And for those of you that have either taken or have taught parasitology course, you know that one of the horrors of this for the students is memorizing these friggin' life cycles for the apicomplexans. I mean, they're all terrible, but the apicomplexans are about the worst. And malaria, you got to teach malaria life cycle if you're going to teach parasitology, and the students hate it. And one of the reasons they hate it is they want to go to med school and they know that they're never going to have to think about malaria, right? And so I was trying to figure out how can I get my students to care about malaria. And at that point, there were these papers coming out on projecting malaria under different climate scenarios. This was from Martins in '99. And so if you do these environmental niches for malaria based on temperature and precipitation, you can get, projections like these are the areas that are suitable for malaria currently. And then you can ask, well, what happens under climate change scenarios? And you can show this to the students and say, listen, you need to study malaria and get the life cycle now because by the time you're a doctor, you're going to be treating patients with malaria. Where you live are the chances. And so I found that to be a really great motivator in my parasitology class. And I was at a ESA meeting about that time, and I ran into John Chase, and I really loved some of the work that he had done on climate and malaria in temporary pools. And so we were chatting about stuff, and I was telling him about this story of how I used, these models to motivate my students. And he said, "Why don't?" He was editor at Ecology Letters at the time, still is. And he said, "Well, why don't you write us a review and synthesis about climate change and parasites?" I'm like, yeah, I've kind of been wanting to do that. I'll give it a try. And so at that point, I was already familiar with a bunch of very influential papers that were, written by friends of mine and colleagues, and pretty much understood the consensus about a warmer world is a sicker world, et cetera. But there were like 300 papers. And so by the time I got to paper 275, which, I started coming across things by folks with different perspectives, like Rogers and Randolph from Oxford. And these guys had been in the parasitology game for a long, long time, and they were just super annoyed with disease ecologists and how they seemed to be oversimplifying a really important question. And so I had to rethink my views about climate change and infectious disease and consider a more nuanced and complicated perspective. And one of the main points that they made, and I also thought was worth repeating, is that when you look at a model, for instance, a malaria transmission model, yeah, temperature is in the center, and disease transmission is quite sensitive to temperature for sure. But when you do the specific modeling and you look at things like, all those parts of the model like development rate change with temperature, right? And what that leads to is this unimodal relationship between R naught and temperature. And speaking to you guys, none of this should be really that surprising to you. But the point is that although R naught might increase with temperature when the temperature is like 25 degrees, at some point you hit the top of the curve. And what this means is that the general prediction is that R naught is going to shift with climate change. Not necessarily, it's not a linear effect. And so what we expect really is for diseases to shift their distribution to higher latitudes and higher altitudes. Anyways, that was a key feature of this paper. It shouldn't surprise anybody nowadays, I suppose. But I sent the paper back to Jonathan Chase, and he sent it out to review. And it was really interesting getting the reviews back because they were all pretty short, and they were, I don't like the flowery California language, and please cite my paper and this and that and blah blah blah. Basically just a couple paragraphs. And then I got down to the bottom, and it says, "And we are rejecting your paper." And I was like, oh, that wasn't where I was expecting this is going. And it reminds me, when you review a paper, everybody's been in this situation. You review a paper, you have the little box that you fill out that the authors are going to read, and then you write what you really feel about the paper to the editor. And so basically, I went back to John. I'm like, I don't get it. The reviews don't seem all that bad. I can accommodate them all. He said, yeah, but the reviewers are really unhappy with this paper. They don't want to see it published. So I was like, oh man, that was a lot. I read 300 papers for nothing. I was kind of pissed off about it. And then I ran into Don Strong, who's the editor of Ecology, at a different meeting, and I was griping with him about ecology letters and how they didn't want to take a risk on this paper, et cetera. And he said, I don't know if you know about Don Strong, but he is just like a magnet to controversy. And he just loved the idea that people were upset about this paper. And he's like, why don't, this is great. Let's just double down on the whole thing. And so he wanted to do a concept and synthesis in ecology. And so, he reviewed the paper, he liked it, and we published it there. But he also invited all these different counter perspectives on the paper, which are also great. And most of these people are, were colleagues and friends of mine. And so that became what I call, at least for me, a really interesting entry into this climate change and infectious disease to think about what the infectious disease ecology community can do by bringing a range of different perspectives. But perhaps the biggest effect that this paper had was that my PhD student, Erin Mordecai, read this paper and she got pretty interested in it because what she pointed out, I said, yeah, Kevin, you're right about this thing about R naught being peaked. But what you're wrong about is the way these models are parameterized, because this is not what the data showed. The data actually show this. In other words, these using these simple linear effects in models is potentially really changing the predictions of these models. So we work together to sort of refine the way that we build R naught projections under temperature scenarios, and in particular, thinking about how to match it up with data. And so when we take sort of the standard malaria model, and Erin redoes it with her little modeling magic, what she finds is the old model's in red, showing a peak transmission of malaria around 34 degrees. But the peak is really about 25 degrees in her model. That's a huge shift. It really changes a lot of the way that you would expect malaria is going to respond to global change. And, it matches what the data say. And so, that's been a real game changer for how Erin has gone on to model a whole range of infectious diseases using these approaches. The point being that not only are these unimodal curves important, but where the peak is important. Getting the peak right is important. And these diseases have different peaks. And so you might expect under a climate change scenario, one particular infectious disease to decline as the other one is increasing. And that's a really important detail to get right in terms of responding to climate change and improving public health. I just gave an example for our malaria model. This is the current niche for malaria transmission in Africa. And then when we apply these models to climate change scenarios, you can see that large parts of Central Africa are just getting too hot for falciparum malaria transmission. And that's not what the previous models would have shown. Okay, so the environmental niche is sensitive to temperature. That's pretty obvious for malaria and a lot of other things. But the other thing I want to mention next is, climate is just one part of the equation, and it's an open question as to whether it's even an important part. And one of the things that Sarah Randolph really emphasized in her work is that you might see what you think is a climate signal, but don't mistake it for an economic one. And so if you build these models and you account statistically for economic growth and the improvement of various conditions, folks have done this. And this is the future change in malaria accounting for economic growth, where blue is contraction and red is expansion. And there is no expansion once you look at the overwhelming effect that economics has on malaria distributions. In fact, if we look at changes in time that have happened already, um, for different types of infectious diseases, 1990 versus 2021, we see what we think is this economic effect. This is a time uh, as the climate has warmed and as other global impacts have increased, diseases are down across the board for in many cases. You can find exceptions to this for a specific disease, but in terms of these broad categories, we are seeing declines, and the expectation that this is due to economic gains overwhelming some of these environmental ones. And what that suggests is that you can't really just study temperature. You can't just study economics. You need to think about ways to put many variables into an analysis. And this is an attempt that we did to look at many different diseases. These are those 24 important human diseases and many different factors. So that's geography, wealth, the extent to which people live in an urban environment, population densities, temperature, rain, forest cover, and biodiversity. And the first column are spatial effects, and the right-hand column are temporal effects. And basically, the main take-home point I want to make from this is that all these diseases respond differently to a range of factors, but there are some general patterns. And if we sort of combine all those things together, what we see is that if we look at the spatial variance on the x-axis and the temporal variance on the y-axis, then the upper right-hand quadrant are things that are consistent in time and space, and the lower left-hand quadrant also consistent in time and space. And basically what this is saying is that infectious diseases have been declining in time and space due to wealth in the proportion of people that live in the urban environment, and to a lesser extent, but also consistently increasing with temperature in time and space. But the effects of biodiversity, population, rainfall, and forestation are inconsistent in time and space, and may be positive, may be negative. But also, and very importantly, there's huge error bars around these things. And so the details are incredibly important if we're going to make predictions here. All right. So I guess my general point here is that, the world is complex. That should be a surprise to nobody, and we need to consider that before making oversimplified conclusions about anything, climate change specifically. But I wanted to say, well, what is the whole point of doing this stuff anyways? And that is that if disease ecologists if society is getting their money's worth from us, then we ought to be able to be informing solutions. And so one of those that we've thought about a lot is the extent to which there are win-win outcomes for conservation and human health. I mean, this is sort of what we would hope to find so that we can improve both the environment and people's lives. And so the idea in this schema is that over time, we might start with a situation where we have pristine environment and good water quality. But then if we do deforestation, the water quality gets bad and we expose people to pathogens. And that would mean that if we do forest restoration, we might get a win-win. We might improve the environment and we might improve people's health. So there's been lots of ideas floated around about how we might do this. And the details of the picture aren't important, except for to say that we can illustrate them with sexy graphics to say, wouldn't it be cool if we could move from these lose-lose situations to these win-win situations by doing things like planting forests or improving conditions for birds or improving water quality, et cetera. And I'm going to end on a downer, but we evaluated 46 of the hypothesized win-wins for conservation and human health, and we asked, how much evidence is there that these things work? And for the most part, not so much, not as much as we might expect, and not as much as we would like. And so basically, for most of them, the evidence for either a health benefit or a conservation benefit is kind of low to medium. And what that means, and I'm really speaking to the younger people in the audience, which is that there's a lot of really important science that needs to be done before we can choose to invest in these particular win-win outcomes. There's a lot more research that needs to happen. And, I really wanted to save some time for questions. And so I'm going to go to my conclusion slide and wrap things up. When and where does global change alter disease risk? And I should put an asterisk there. These are my opinions and the things that I want to emphasize. You can emphasize your own conclusions if you wish, but I think it's pretty undeniable that most human impacts are in the global south, but most research is not. And so, we might want to rethink where we direct our attention and effort. Um, Surprising, I think, from a disease ecology perspective, most human diseases have declined despite of global change. And I think that doesn't necessarily mean that these environmental effects are not important. It just means that they are not dominating the story at this point, that other things are likely important, at least at the broad level. And one of the reasons that this may be the case is that stress increases some diseases and it decreases others, and the details matter. You need to know your disease and you need to know your stress, or you're not going to be able to make a good prediction about how stress affects infectious disease. Um, and similarly, food web simplification can impact parasites because parasites depend on complex food webs, but it can also increase disease indirectly in some cases, as that example I gave with the lobsters or with human schistosomiasis. So food webs are also important to understand, I think. Biodiversity increases some diseases, decreases others. And again, the details matter and require, I think, very careful research before overgeneralizing one way or the other, independent of how good it makes us feel to have a particular outcome. Similarly, warming may shift vector-borne diseases higher in altitude and latitude, but even if it has those effects, the economics can overshadow the ecology. And so we do need, I think, to pay attention to all aspects, even outside our comfort zone, with respect to, the future of how global change is going to affect human diseases. Because global change is not just about climate change. It's not just about biodiversity loss. It's actually as much about economics as anything else. And interestingly enough, the world used to spend a lot of money on combating infectious diseases in the global south, and we are changing that. We have decided as a society to change that quite a lot. That may lead to changes in people's health around the world that may overshadow these ecological effects. And then finally, I want to say even as attractive as win-win solutions are, and as much as I hope that they work, most are untested. We were aiming for a win-win solution in Senegal with restoring native prawns to that food web and benefiting people's health with schistosomiasis. But we decided we needed to test it before doing it, and our results were encouraging in some ways and not in others. And with that, I'm going to wrap up. And if people have points that they would like to make or questions that I can answer, people can put it in the chat. But I would much prefer if they raise their hands and they could ask it in person. And I will say that thank you, Paul, for clapping. Really appreciate everybody being here. I do have a question from Jesse in France about the prawns in Senegal. Do people eat the prawns and can cercaria transmit that way? So the answer is yes, people eat the prawns, and no, you can't get infected by eating the prawns. That's an important detail of the life cycle. One of the reasons we had high hopes for this approach was that the villagers could farm the prawns and not only get some protein, but they could sell the prawns and get some economic benefits and get some health benefits and get some river restoration benefits. It was a win, 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 win. And there's not a lot of those on the menu. God, I wish that there was ways we could make that work.
Thank you, Kevin. Any other questions? Yes.
Hi, Kevin. Great talk. Thank you. So I loved all that you presented. And one of the things that I come back to a lot is that parasitism is a pretty broad trophic strategy, like predation. And probably there will be some winners and some losers during environmental and global change. And I like to think a lot about going into like New York City, and maybe there you see rats and pigeons and raccoons. So I'm really curious, among the different trophic strategies and host interactions within the parasites, who do you see as kind of the pigeons and the rats of the future?
Yeah. Right. So, I mean, I think that what I am most worried about are um, simple life cycle pathogens that are fairly generalist in terms of the host that they can use. I think those are the ones that are, going to be. There's just fewer opportunities for intervening and dealing with those. So. Um, I don't know if this is a good example or not, but West Nile virus, you can deal with it in humans, but getting rid of it in rodents isn't going to happen, right? It's generalist. It doesn't require a vector. You can't do vector control to get rid of a number of things. So, and I'm not sure if I'm answering your question about who the rats are, but on the other hand, if you look at the impacts to human health and who are causing those things, that I think is a good way to start as well. Like, um, HIV and tuberculosis are EID people don't think about those diseases. Maybe we should. I don't know. Maybe there's an intersection with ecology with those. Malaria certainly and schistosomiasis for the parasitic diseases are way up there on the list, and yet and certainly people do study those, but maybe they deserve outsized effort for those reasons. But in terms of the ones that are likely to respond positively to global change, that's a key research question. And I think understanding life cycles is one of the elements there, and I'm sure you would agree with that. I have a comment from Jesse that SARS and COVID is pretty generalist. That's true. That's not helping us, I'm sure. And then I have a question from Noah. Great presentation. Thank you, Noah. I work on human factors, schistosomiasis, and malaria in my master's. Is there anyone we could work with for my PhD? I'm actively seeking to advance the study. I can share details of the master's study. Noah, I am too old to take new students, so I'm going to pass that buck. But I will say that Noah, that this is a really, firstly, there is certainly research activity going on there right now. The U.S. Is a complicated place, I think, to be doing work on malaria or schistosomiasis in terms of the funding that's going to be available to work outside of the country. And so I would just say that's uncertain. Maybe looking in other places like Europe would be might be more likely. Yeah, good luck with it. Please, please, please. I'm just begging people to do exactly what you're asking to do. So I do hope that you find success with that. I have a question from Ann Clark. Great talk, and the scope underlines how one could teach all of biology using a disease organism perspective, leading to forays into basic concepts in EEB. I agree with you, and what's interesting is that in ecology courses, diseases, at least historically, got very little traction, and yet they're so illustrative of so many concepts. Now that has changed, and I think that I mean I don't teach undergrads, but I think I would imagine that in a typical ecology course, infectious diseases are getting a little bit more love than they used to. So hopefully that's the point. Question from Joseph for point two. Is this decrease seen when observing disability adjusted life years versus considering the incidence of cases or cases of disease also broadly decreasing? Aha, yes, good point. Yeah, so I love using the disability life years as a measure when I'm comparing multiple diseases in the same study. It may not, if you're just looking at malaria, then looking at, incidence or prevalence or whatever might be what you would stick with. But if you're comparing malaria and schistosomiasis and HIV and everything else, you got to go with the dailies. I think for those analyses, at least that's what I've done. And cases could easily have a different pattern if medical interventions are reducing the disease impacts of an infection, for instance. And so that's a very good point. Okay.
Hey,
And
Kevin, I've
then,
got a question.
yeah, go
Sorry,
ahead, Jamie.
weirdly not put my hand up. Amazing talk. Thank you. I'm curious. I've heard the idea with regard to win-win solutions, that the evidence threshold doesn't need to be as high. If you can win in two axes, then you don't need to have as high a degree of confidence as you do, say, if there's a trade-off. I'd love to hear your reaction to that.
Yeah, let me. I totally agree with you. Any win is great, right? And so if you could get high in one column, then that's probably good enough reason to implement that, assuming that there aren't some hidden negative effects that there might, and in some of these cases there could be, I suppose. But the high column is pretty sparsely populated too. So but you're absolutely right, that you don't need both axes to move forward from a human societal perspective.
So I haven't digested that. It's interesting that the high evidence quality is worse for human health outcomes than for conservation. Do you think, is that a consequence of the nature of these win-win solutions and who's been thinking about them for longer?
Well, it certainly doesn't include lots of human health interventions that aren't targeted toward conservation, which have high evidence that they're successful, vaccination rates, right?
Yeah.
So those aren't on here necessarily. These are just ones that have stated up front, this is a win-win, we should do it. And then we've gone through and said, how much evidence is there that it would work? Is this backed with data, or is it just a good idea? And to your point, Jamie, I believe that the reason that the conservation evidence has higher quality is that these are being pitched from a conservation perspective almost entirely. Basically, people that want to see a conservation outcome are throwing in human health as a side benefit to their proposal. So they have more evidence for their conservation than the human health is more speculative. Does that sort of jibe with what your expectation is?
That's where I was sort of inching towards. So yeah, thanks for clarifying. I'll let others ask their questions, but thanks.
Okay.
Maggie, would you like to go?
Yeah, thanks. I really enjoyed this, so thank you. And I was just curious, you mentioned early on the idea that particular parasite characteristics like life cycle complexity or like specificity to certain hosts could help us predict parasite responses to host biodiversity changes. And I was wondering for these like very diverse parasite responses to stressors, how you think about, or if you think about maybe those characteristics or other parasite characteristics that could help us understand maybe which parasites are likely to respond one way or another to like thermal stress or to pollution.
Yeah, that's a great question, and the answer is yes. And just as for an example, for thermal responses, a species that has an ectotherm host is going to be much more sensitive to temperature change than one that lives in an endotherm. A pathogen that has a environmental stage is going to be much more sensitive to environmental effects than one that does not have an environmental stage. And so, for instance, influenza is a really good example. That virion that's sitting out in the environment waiting for you to contact it breaks down a lot faster when it's warm than when it's cold. And so it's one reason why the flu tends to get, is more prevalent in the wintertime than the summertime, just because that infective stage has a longer shelf life. And so those are the two things for sensitivity to thermal that we know are important. And then, the other things like pollution and so forth, you can sort of make a list of what makes a parasite or infectious disease sensitive to these other stressors. It really has to do with their exposure and intimacy with that particular stressor.
Melody, did you want to ask your question?
Yeah, if we still have the time for it,
Sure.
I don't want to run up the clock.
Yeah.
Thank you, Dr. Lafferty, for giving the talk today. I'm a doctoral student right now, so I'm not as far along and I haven't built the intuition for a lot of these things yet. But taking a little bit of a step back on the balance of like global human development versus the impacts on infectious disease, I've personally encountered a lot of trouble sort of balancing or not balancing or of including both the human dimension and the disease dimension of it, because I think people hear parasite and they don't really think about parasite conservation and parasite diversity as a good thing, and that gut instinct to sort of shy away from that. How have you historically managed like communication of the need to balance sort of what's best for humans versus what's best for sort of ecosystem writ large?
Yeah, that's a great question. I think about it a lot. One of the reasons I had that slide showing which parasites are problems kind of relates to that, which is to say, um, I am certainly on the list of people who promote things like parasite conservation, but I always make a really strong point to say we're not talking about conserving parasites that, make little children sick. That's not what we're after here. We're talking about the vast majority of species that are not problems from a human perspective, and we can think about conserving those. So that's probably, I would say, one motivation to think carefully about whether a parasite is a problem or not. And it can get deeper than that, but it's an important question. And yeah, parasites are part of diversity, but I also am quite careful to not say that we need to preserve malaria that kills tons of babies in Africa just because it doesn't affect me. You know what I mean?
Absolutely. Thank you so much.
Sure. Costanza?
Yes, thank you for this talk, Dr. Lafferty. It was really great. I'm talking as a wildlife disease ecologist, so much harder for us to get the message across without the human components. I'm specifically specialized in sea turtles cancer. But I wanted to ask you, because working in the One Health environment, from your experience, do you reckon that COVID has served as a wake-up call outside of the scientific community for society in terms of global change, occasionally equal diseases, or if we simply put a band-aid on it as a society and things will continue business as usual? And if so, what are the best approaches as scientists to avoid that moving forward?
I don't think there's ever been an infectious disease in modern times that such a huge fraction of the globe, has thought about. It's affected daily lives, right? And so, on the one hand, you would think that would be a great opportunity to have more increased awareness, et cetera, et cetera. But I actually don't think that, at least in our field, that COVID has helped clarify or motivate, things the way that you might hope. It's an interesting question that you raise. I don't see it. For any number of reasons, it would be a very long discussion, including just the fog of how COVID arose and how it spreads and all those other things, and whether we learn general things from it or not, or whether it's so different and unique that it's difficult to generalize from. I don't know personally, but it's a really excellent question.
Thank you very much.
I have a question in the chat from Itamar. I think it's safe to assume a lot of the host parasite interactions at the species level are missing. I'm curious, how do you think this affects or creates biases of our predictions to global change effects on disease risk? And so I'll rephrase the question in a way that makes it easier for me to answer, which is that the scientific attention to diseases is very idiosyncratic in terms of where an individual researcher wants to spend and focus their energy. And so it's not necessarily, it is quite biased. Like, for instance, I mean, we don't cover the full range of taxonomic breadth of infectious organisms or hosts. We tend to focus on things that affect wildlife or humans or other things that we care about, and that's not a random subset. We also tend to focus on systems that are tractable. If you can work on it in the lab, then you're going to be much more likely to get an NIH grant than if you can't. If it infects people that live in your home country, you're more likely to be motivated to study that or to get money to study it. And so, if your country doesn't have a major research program that's funded, you may not be getting the benefits from the science that you need in that regard. And so I'm not sure if that's exactly what your question is, but yeah, we are not, nobody is going out in a systematic manner and studying host parasite interactions, in that way. They're biases. I'm not saying the biases are good or bad. They're just they're a product of the way that the science business model works.
Maybe what we'll do at this point is, thanks, Kevin, for your talk. I know Kevin can stay on maybe a little bit longer, but maybe for the purposes of the video, we can sort of stop the video and whatnot. And then, like, if people want to hang on for a couple more questions, feel free to do that.